Hello, thank you all for being here. Before I start telling you about some brand new population genomic results we have testing the idea that character displacement can initiate speciation, I would first like to acknowledge my collaborators, Corinne and David Fennick from the University of North Carolina at Chapel Hill. And I especially want to acknowledge my postdoc, Heidi Sears, who unfortunately couldn't come to the meeting this week, but actually did most of the work that I'm going to be telling you about, so I want to give credit to her. So first, what is character displacement? So when you have two species, closely related species, that co-occur, they often will compete for resources or even mates. And that is depicted here by these overlapping distributions of resource use or mating traits. Now, you can imagine that individuals of either species that are less similar to the other species should experience less competition and have lower risk of hybridization with the other species. And this should give a fitness advantage to individuals on the tails of these distributions. Over time, this can result in divergence between the two species where they co-occur in those resource use or mating traits. So this process here is called character displacement, and it has been long recognized as a mechanism for finalizing the speciation process and for enhancing species differences. Now, how does it initiate speciation? If we consider a situation where populations of each species sometimes co-occur, where the two species sometimes co-occur, and there are other populations where each species occurs alone, that's how we can get this happening. So if we look up here, these are populations where each species occurs by itself. We'll call those allopatric populations. They are not experiencing character displacement. However, where the two species co-occur, they undergo character displacement, as I just described, and you get this divergence. So now if we focus on comparing populations of a single species, let's talk about species 1 here, between regions where they co-occur with their competitor species and regions where they don't, you can see that because of this character displacement here in sympatry, we now have divergence between populations of species 1 in allopatric versus sympatry. So we have divergence in these resource use or mating traits, and it's possible that that can lead to reproductive isolation. For instance, individuals from the two different selective environments may no longer recognize each other as potential mates. And if they do mate, population hybrids between these two selective environments may be selected against. Okay? So both of those mechanisms could reduce gene flow, potentially to the point where we get incipient speciation between these two selective environments. So we've been testing this idea in the spadefoot toad system. So here I have two species of spadefoot toads pictured, and their ranges here. This is the range of Speabomophrons and the range of Speamultiplicata. And you can see in the southwestern United States, there's a large area of potential sympatry where the ranges overlap. We're going to be focusing for the rest of the talk on this region here, which is in southeastern Arizona and southwestern New Mexico on the corner or the edge of sympatry. In this region, at low elevations, there are ponds where the two species co-occur. Their tadpoles compete for resources, and the adults occasionally hybridize. Previous work has shown that in those sympatric populations, the two species have undergone character displacement in tadpole resource use traits and in mating call rates and mating preferences. However, if we go just a little bit up in elevation to some very nearby ponds, we will find allopatric populations. So populations where only this species is present. So here they're undergoing character displacement, and here there is no character displacement for Speamultiplicata. So we have evidence that if we're comparing those allopatric and sympatric populations of Speamultiplicata, premating reproductive isolation has evolved. When we collect females from allopatric ponds, which are pictured here in red, and from sympatric ponds, which are pictured here in blue, we bring them into the lab, and we play mating calls from both allopatric and sympatric males to these females. The females from sympatry prefer the call rates of male that's typical of males from sympatry, their own selective environment. On the other hand, females collected from allopatry prefer a faster call, which is more typical of males from allopatry. So females from the two selective environments are less likely to mate with male from the opposite selective environment. Likewise, we actually have evidence that there is postzygotic reproductive isolation between populations in these two selective environments as well. We collected adults from these two selective environments, mated them in the lab, and came up with groups of tadpoles that either shared parents from the same selective environment or had parents from different selective environments. We took those tadpoles and raised them with limited resources, either by themselves or in competition with other tadpoles, and asked whether tadpoles that had parents from the same selective environment performed better in those situations than tadpoles with parents from different environments. And indeed, that's what we found. Here, on the y-axis here, this is the difference in body size, a proxy for fitness, between tadpoles that had parents of the same selective environment type and parents from different. And you can see that there was a performance advantage for those offspring that had parents from the same selective environment. Okay, so even if there is mating between parents from sympatry and allopatry, their tadpoles are potentially selected against because of competition. If we look in the field for evidence of genetic divergence, we find that based on 8 microsatellite markers, there is some structure that suggests that there could be some genetic divergence between these population selective environments. The allopatric populations, these red ones here, are more likely to belong to this red genetic cluster, and the sympatric populations, the blue ones, individuals from those populations are more likely to belong to the blue genetic cluster. However, these results are based on only a handful of molecular markers, and so we really have a lot of questions left about the genetic differentiation between these selective environments. So if our overall research question is whether or not character displacement is initiating speciation between sympatric and allopatric populations of Spia multiplicata, we might ask whether these populations are showing genomic differentiation. And if they are, to what extent is that differentiation present? So that's what this talk is about. So to answer those questions, our collaborators collected tissue samples from adult Spia multiplicata from breeding aggregations that look like this, from a couple of allopatric populations, 3 sympatric populations, and then also so that we could examine the relative influence of geographic distance as well as difference in selective environment. We also have tissue samples from populations that are allopatric, but quite far away from our other focal populations. So with these tissue samples, we performed double digest RAD sequencing. We digested the genomic DNA with 2 restriction enzymes and selected for approximately 380 pair link RAD tags. We then sequenced these fragments on 2 lanes of Illumina sequencing and ended up with 558 million raw sequencing reads. We then assembled the loci using a STAX pipeline and filtered the loci so that they were present in at least 40% of individuals and at least 2 or 3 population groups. After all that, we had 144 individuals, and we had over 14,000 single nucleotide polymorphisms. Okay, so we wanted to know whether there was any genomic differentiation, first of all, and if there was genomic differentiation, whether it was associated with those selective environments. So here I have a histogram showing global FSTs across those 14,400 SNPs. You can see that the range is quite large from 0 to 0.53, but the vast majority of loci are clustered around 0. So the overall level of genomic differentiation is quite low. We did a discriminant analysis of principal components to explore whether or not there was any structure related to these selective environments. And we see that most of the structure is related to the geographic separation between those distant allopatric sites and the sympatric and allopatric sites. On this axis, we have some weak differentiation between those sympatric and allopatric sites that are nearby to each other. When we do the same thing, but look only at the top, the 90th percentile and above loci, so the higher FST loci, we see slightly more clear differentiation between our sympatric and allopatric population types. So this is potentially consistent with very early stage speciation or potentially speciation with quite a lot of ongoing gene flow. We also used Bayescan to identify outlier loci that are potentially under divergent selection or linked to selected loci. When we did comparisons between our distant groups versus our sympatric groups, that's our most distant comparison because they're geographically separated and they're different selective environments. We had 11 outlier loci. When we compared the distant allopatric versus the allopatric, so only differing really in geography and not so much selective environment, we had 8 outlier loci. And then, as we expected, the fewest number of outlier loci were 3, and that was between these sympatric and allopatric populations. So we have a lot more that we're doing. These are very recent results. We're going to separate all of our population groups into the individual populations. We're going to do cluster analyses, a more quantitative assessment of the relative contributions of geographic distance and selective differences on that genomic structure, and also use several other methods for typing outlier loci and ask whether or not the outliers that are identified using these different methods overlap at all, which would be strong evidence for selection. And then finally, screen for potential introgression in those sympatric populations, because remember, speed omicrons and speed multiplicata sometimes do hybridize, and that could influence some of the differentiation that we're considering. So I hope I've convinced you that the spadefoot toads are a potential illustration of how character displacement can initiate speciation. There are multiple forms of reproductive isolation. We do have some genomic differentiation associated with selective environment. However, only a small portion of the genome shows differentiation, and we do have a couple of outlier loci that we'd like to investigate more as being potentially under divergent selection. So with that, I would again like to acknowledge Heidi and let you know that she's on the job market, so if anybody knows of any openings, let me know and I'll pass on the info. Thanks.
Was this clear that the ecological differentiation occurs in sympatric versus allopatric? Do you have any data about how different ecologically by the allopatric population that are hybrids with allopatric and then functional as you are presented?
Can you summarize the question? Okay, so the question is, are the hybrids, so you mean the population hybrids that are processed?
You can make F1s. The allopatric population, are they perform as easily as the F1 sympatric?
Okay, so F1s that are processed between allopatric?
Far allopatric.
Yes, are far allopatry times allopatry. If you cross the far allopatry with the allopatric toads versus close allopatry with sympatric toads?
Yeah, so I want to know if the ecological divergence happened in allopatry or in sympatric. We have evidence that the main behavior was changed in sympatric. And I don't see any evidence that ecology has changed in sympatric.
Okay, so we haven't crossed the toads from the distant allopatric populations with the allopatric or with the sympatric populations. Yes, so I cannot definitely answer that question. Okay, I think that's it. Thank you very much.